and welcome to the Absolute Recap Biology Edition, where we aim to maximize your understanding and minimize your need for memorization. Each episode will recap content, skills, and test-taking tips to help you succeed in May. I'm your host, Melanie Kingett, and your recap starts now. Hi, and welcome to the Absolute Recap Biology Edition. Today's episode will recap transcription versus replication. Let's zoom out. We're in Unit 6, Gene Expression and Regulation, Topic 6.2 and 6.3. Our big idea is information storage and transmission. Let's think about this one. How does the structure of DNA influence the processes of replication and transcription?
I'll show you how.
DNA is an anti-parallel double helix with a sugar phosphate backbone and nitrogenous bases joined through hydrogen bonds. The meat and potatoes of DNA is with the sequence of the bases, the A's, T's, C's, and G's. In order to replicate or transcribe the sequence, we will need to break the hydrogen bonds to access the code. Additionally, DNA has directionality, with enzymes interacting in a singular direction. Let's zoom in. When is this all happening? Recall that eukaryotic cells go through phases of the cell cycle. Interphase with G1, S, and G2 before nuclear division through mitosis and cellular division of cytokinesis. Both processes of replication and transcription occur during interphase. DNA replication occurs once during the S phase and preparation for division. After all, the S of interphase stands for synthesis, the time when DNA in loose chromatin form will be copied with the assistance of enzymes within the nucleus. Transcription also occurs in the nucleus. However, it happens hundreds, if not thousands of times throughout a cell's lifespan during G1 and G2. Transcription is the process of reading a DNA strand and forming an mRNA transcript. It occurs every time the cell needs to synthesize proteins, as it is the first step of the central dogma. Quantity matters. DNA replication is a semi-conservative process that copies an entire genome. After all, we want to make sure that we have identical information in the new daughter cells. Each original half of the parent strand serves as a template for new complementary nucleotides to be synthesized. In other words, half of the original strand is conserved or saved during replication. In transcription, very small portions of the genome are transcribed from one template strand, not both. This template strand of DNA is also known as the non-coding, minus, or antisense strand. We only need to have the sequence relevant to the gene being read. The mRNA transcript formed is single-stranded and entirely new. Directionality. It is so important. Just as English reads the left side of the paper to the right side of the paper, replication and transcription are direction-specific processes. To understand why, let's look at a nucleotide more closely, the monomer of nucleic acids. A nucleotide is composed of a pentose sugar, phosphate group, and nitrogenous base. The pentose sugar contains 5 carbons formed in a ring. Imagine the nucleotide like a kindergarten sketch. You have the house, which is the sugar, the sun above the house, which is a phosphate group, and a garage, the nitrogenous base. In DNA, this is a deoxyribose sugar and nitrogenous bases A, T, C, and G. Whereas in RNA, it's a ribose sugar and bases A, U, C, and G. Looking more closely at the pentose sugar, we can identify the locations of the 5 carbons.
Hello, AP.
So we'll just leave this blank for now, and I'll come back and say the subject name here.
Student, I know that you are currently preparing for your AP
The subject name.
exam, but are you taking more than one AP course? Maybe you want to get a jump on courses you plan on taking in the future? Do you think that you're better than me? Is that what you think? Because I'll have you know that Along with the Absolute Recap,
the subject name
edition, we have podcasts and study guides for biology, chemistry, music theory, physics 1, and U.S. government. So if you think you're better than me, if you are currently taking or plan on taking more than one AP course, visit www.TheApsoluteRecap.com. That's www.TheApsoluteRecap.com. And click on the courses link or search The Absolute Recap on any podcasting platform. And now, back to The Absolute Recap,
the subject name
edition.
I'm going to stick with the house analogy for a few more minutes. Starting my viewpoint at the peak of the roof, I see an oxygen. No carbon there. Moving my eyes down the roof line and to the right, I land at the 1' carbon. This first carbon forms a covalent bond with a nitrogenous base. Continuing around the pentose ring in a clockwise fashion, I count my carbons. 2' and 3' along the bottom, 4' at the left roof corner, and then up the chimney to carbon number 5. The 5' carbon connects to the phosphate group within a nucleotide, while the 3' carbon connects to the phosphate group of the adjacent nucleotide, forming a sugar phosphate backbone. Okay, great. I can count carbons. How is that useful?
Well, I must say, that's great.
Well, it's important to be able to distinguish the orientation of the molecule. For reference, the 5' carbon and the oxygen roof of the pentose sugar point in the same direction. Second, DNA is anti-parallel, with each strand running in opposite directions, 5' to 3' on one half and 3' to 5' on the other. Third, enzymes are direction-specific. The enzymes responsible for synthesizing either the new DNA strand or the RNA transcript are called polymerases, literally polymer makers. Each reads the template strand in the 3' to 5' direction, while it synthesizes complementary nucleotides in the 5' to 3' direction. DNA polymerase binds to a short RNA primer and proofreads its own work as it synthesizes. On the leading strand, this synthesis is continuous in one piece, as DNA polymerase follows the replication fork in the same direction that the enzyme helicase unwinds. But remember, DNA strands are anti-parallel, so DNA polymerase synthesizes away from helicase on the lagging strand in short fragments. RNA polymerase and other transcription factors do not require primers, but rather recognize a promoter sequence on the template strand, called the TATA box in eukaryotes. While the information is not proofread, there will be mRNA processing following transcription before translation in eukaryotes. Since DNA is organized differently in prokaryotes and eukaryotes, it will have different strategies for replication and transcription. Prokaryotic DNA is organized in a singular, circular chromosome and has only one origin site of replication. Eukaryotic chromosomes are linear, contain multiple origin sites of replication, and often lose the ends of their chromosomes, called telomeres, with each replication. Eukaryotic transcription is followed by mRNA processing, while prokaryotic transcription overlaps with translation, so no RNA modification occurs. Regardless of the process, Chargaff's rules are still in effect. Guanine is complementary to cytosine, and adenine is complementary to thymine or uracil. Quick practice. If the DNA template was 3' to 5', TACGCA, what is the DNA complement and mRNA transcript? DNA complement is 5' ATG CGT 3', and mRNA transcript is 5' AUG CUT 3'. What? Those are so similar.
I see what you mean.
Yep, the non-template strand of DNA is often referred to as the coding strand. Just swap uracil for thymine. Time for unit connections. Unit 1, Chemistry of Life, with nucleic acids and protein structure. Unit 3, Cellular Energetics, with enzymes. And even Unit 4, because transcription and translation is often the result of cell signaling. All right, what about the exam? Don't get tripped up by directionality, as that may be a feature in questions. It's unlikely that you will be asked to replicate or transcribe a sequence outright, but rather discuss cause and effect or disruption to a process. Also, you may need to distinguish between gene expression in prokaryotes and eukaryotes. Oh, and review the lac operon. We will go over this in our next episode. To recap, DNA is all the rage during the processes of replication and transcription. Compare and contrast for timing, directionality, enzymes, nucleotides, cell types, and nitrogenous base pairings. Coming up next on the APsolute Biology Edition, crafting a gene. Today's question of the day is about nitrogenous bases. Which nitrogenous bases are classified as purines? For the answer to the question of the day, please follow us on Instagram at TheApsoluteRecap. That's the APsolute recap. Check out our website, TheApsoluteRecap.com, for episode schedules, study guides, virtual tutoring, and to sign up for our virtual classroom. The Absolute Recap is produced by Brad Kingett with music by Zach Caruso. Today's episode was written by me, Melanie Kingett. Thanks for subscribing, and don't forget to rate and review wherever you get podcasts. Time's up, pencils down. Thank you for listening to the Absolute Recap Biology Edition. AP is a registered trademark of the College Board. Copyright 2020, Absolute Recap LLC, all rights reserved.